Hello, fellow biologists. We are now in part B of chapter 4 on translation, and this will be the second and last video that we will have in order to cover the section on translation. So this should be the conclusion of this segment on translation. So again, don't worry about the earlier chapter, or earlier section 4.1, or the sections after this. Now here in part B, we are going to focus on um, the process of protein synthesis now that we have become familiarized with the various parts, working parts of the molecular machine. So the process of translation is divided into three separate processes. The first is initiation, whereby the initiator AUG codon is recognized and methionine is established as the first amino acid in the new polypeptide chain. And this is true whether you're dealing with prokaryotes or also in eukaryotes. And the second stage is called the elongation. Once initiation process happens and methionine has been incorporated as the first amino acid, there would be a process called elongation where successive amino acids are being added to the growing chain, okay, with the sequence of the amino acids being determined by the respective codon of the incoming amino acids, but they have to be complementary in a codon-anticodon interaction principle. And then the third process is, of course, termination, where the addition of amino acids would stop by the recognition of a stop codon and the subsequent polypeptide chain being released from the ribosome. So let's now look at each of these stage in sequence and see what personalities or functional parts are involved. Okay, so let's have a look at initiation first in eukaryotes. Okay, to be sure, although elongation and termination are very similar in prokaryotes and eukaryotes, their translation initiation mechanisms differ from each other. Okay, and you must remember that in eukaryotes, the transcription of the messenger RNA leads to processing so that now you have a mature RNA that gets exported into the cytoplasm, and that is where translation happens. And each mRNA that gets exported into the cytoplasm are mature mRNA because they have undergone processing, and they always encode a single polypeptide chain. That's why eukaryotic mRNAs are called monocistronic mRNAs, meaning they just code one polypeptide, or they only have one open reading frame, or ORF for short. Okay, so please remember that, it's very important. Now, in the process of initiation of translation in eukaryotes, there is a very important structure in the messenger RNA that is very important, and that is the 5' cap structure. It's a very specialized structure at the end of the mRNA that is not encoded by the DNA but is post-transcriptionally added during the processing event, okay, as you learned in chapter 3. And this 5' cap structure, as we will see later on, binds initiation factors that allow the ribosomes to be recruited. Okay, and the ribosome, particularly as we will see later on, the details will be coming shortly, but the small ribosomal subunit is going to scan until it reaches the AUG codon, the first AUG codon, which codes for the initiating methionine. And then the large subunit would join at this point in order to begin translation. Now, it is important to point out that the scanning process proceeds in a 5' to 3' direction until the first AUG is encountered. So it is the 5' cap structure in the messenger RNA that actually directs the ribosome to scan in this direction from 5' to 3'. And the initiation of translation begins the synthesis or the formation of the growing polypeptide chain. First, the first amino acid uh, will have what we call as the amino terminus of the growing polypeptide chain. And as elongation and termination occurs, subsequently, the last amino acid that gets incorporated will then terminate into what we call as the carboxyl terminus of the polypeptide. Now let's look at initiation in prokaryotes. They are, again, as I said, very different because of two important architectural differences. So first is, of course, the fact that in prokaryotes, um, there is no physical separation between transcription and translation because there is no nuclear membrane that separates the two processes. And in fact, it's important to remember that in prokaryotes, transcription and translation, okay, can occur concurrently. In other words, a transcribing mRNA can immediately be recruited to translation even while transcription has not yet been completed. So we call that as co-translational process. Okay, so the transcription and translation in prokaryotes are actually coupled events. Okay, and that second difference is the fact that the mRNA in prokaryotes are what we call polycistronic. Okay, so what that means is that it has more than one, greater than one open reading frame, meaning that it encodes more than one polypeptide as illustrated here. So in this particular example, we actually have a polycistronic mRNA that has or that encodes 3 different polypeptides. Each of these polypeptide have their own AUG codon, represented in yellow and in green. Okay, and the purple ones are indicates the stop codon. Okay, so the start codon and the stop codon defines what we call as the open reading frame, which delineates the region that produces the polypeptide. Okay, so in a polycistronic mRNA, the initiation complex is actually formed at one or more internal sequences present in the mRNA, and they are known as Shine-Dalgarno sequence, named after the scientists that first detected this conserved sequence. So each of these polycistronic mRNA contain in each of the open reading frame unit this yellow sequence called Shine-Dalgarno sequence. And that is where the ribosomes bind, okay, and they're all found internally so that translation can actually be initiated simultaneously at internal sites. Okay, so in contrast to eukaryotes. So again, Just like in eukaryotes, however, the ribosome scans 5' to 3' direction until it counters the first AUG in mRNA. So this is a common property of both prokaryotes and eukaryotes. Okay, so I would like to emphasize the point that in prokaryotes, they don't have a cap structure. Instead, they have the Shine-Dalgarno sequence where the ribosome gets recruited. So what are the roles of the initiation factors in this process? So there are actually two ways by which initiation factors contribute. There's very specific and important roles. In the first step is that there is a certain group of initiation factors that bring up a transfer tRNA charged with methionine called as we know already, the tRNA methionine. This is the initiator tRNA into the small, okay, ribosomal subunit as indicated over here. Okay, so one of the functions then of initiation factors is to enable the small ribosomal subunit to bind the initiator tRNA. So that's a very important step. That's step number one. Step number two is the messenger RNA. On the other hand, we're using here the eukaryotic mRNA as an example. As I said earlier, the messenger RNA's cap structure has to also bind a different type of initiation factors. And they are essentially called cap binding protein. Okay, and as we will see, these or proteins, as we will see, this complex called cap binding proteins must bind the messenger RNA in order to allow the messenger RNA to bind the small ribosomal subunit. So these are two coordinated steps. A distinct group of initiation factors helping prepare the small ribosomal subunit by recruiting tRNA on it. And then the second step, which is the preparation of the mRNA by the binding of another set of initiation factors called cap binding proteins in the cap structure, which then allows the mRNA and the small ribosomal subunit with bound initiator tRNA, charged tRNA, to form complex together. So in step three, therefore, the initiation factors recruit the small subunit of the ribosome carrying initiation factors with tRNA from step one. And then four, the initiation complex then starts moving along the mRNA, okay, which technically we call as the scanning process, okay, until it encounters the first AUG triplet. And the position of the first AUG establishes the translational reading frame, as we have already indicated in previous slides. So this is the role of the initiation factors in the initiation process. Very important. So they all coordinate these specific steps together. Next, upon reaching the AUG, and this is the very critical step, this is where the first decoding process happens. When the small ribosomal subunit arrives and encounters the first AUG codon, the anticodon UAC in the initiator tRNA that carries the methionine, okay, forms a anticodon interaction complex, right? The hydrogen bonds between these two, the codon and the anticodon happens, and that is where the first decoding event take place. They have to be correctly complementary to each other in order for this decoding to happen normally. And once this happens, it actually triggers the small ribosomal subunit to join to a large subunit. Okay, this large subunit here gets recruited once the first AUG codon is encountered by the small ribosomal subunit in conjunction with the initiator tRNA methionine. So the important event here during this process of large ribosomal subunit joining is that the large ribosomal subunit binds the tRNA at the P site at a very specific position. So this initiator tRNA carrying methionine must bind at the P site. And the reason for that is it orients and positions all of the necessary binding sites for the elongation process to happen. Okay, and the reason is that if it's bound, if the tRNA initiator tRNA is bound at the P site, then the MTA site is now able to receive the second tRNA, the incoming second tRNA in this particular case, tRNA that carries a valine, and that's because this tRNA has an anticodon, CAU, that is complementary with the codon GUA. So when this happens, the second incoming tRNA charged with valine allows the ribosome to have an anticodon codon interaction, and the second decoding event happens. And upon doing this, the initiation factors are released, and this now prepares the or allows the ribosome to enter into the next stage of translation, which is called elongation. Now, elongation commences with the formation of the first peptide bond between the two amino acids, and this is how it takes place. Once the new tRNA charged with the valine is in place in the A site, a what we call the coupled reaction takes place. And what this means is that the covalent bond that connects the methionine RNA to its tRNA is broken, but is transferred to the amino group of the next amino acid, which is valine. So the bond here gets transferred over here. And since it's a transfer of bond, there is no loss of energy. It's an equivalent reaction, but it's a directed kind of reaction, directed by the large ribosomal subunits. And this process of coupled reaction generates the first peptide bond. Notice that this happens, okay, in the region of the P sites. So effectively, what happens is that the new peptide, in this case now, you have a dipeptide because you have two amino acids linked together. They are now attached to the tRNA in the A sites, leaving the tRNA in the P site with no amino acid anymore, and therefore is considered uncharged. Okay. Now, the formation of the peptide bond, this is a very important and amazing finding. Requires multiple proteins in the large subunit. Okay. However, it has been discovered quite recently that the RNA itself in the large subunit is the actual enzyme or the actual catalyst. It's not the proteins. So this is a most surprising finding that demonstrate that the RNA can actually act as an enzyme, and therefore called technically a ribozyme. And this finding has led to the discoverer of this phenomenon to win the Nobel Prize, because of the evolutionary implication of how translation has first emerged or originated. So, according to the RNA, so-called RNA world hypothesis, okay, um, that's the transfer of information in order to build complex molecules has actually been mediated originally not by proteins but by RNA. Okay, and so this is a key evidence that is consistent with this hypothesis. So based on those interesting ideas that you have learned, so why don't you try to answer this riff question? Peptide bond formation is catalyzed by the enzymatic activity of an RNA molecule. Is that true or is that false? Okay, so the next stage after the formation of the first peptide bond is the movement of the ribosome. Now, in your textbook, this is recognized as the formal start of elongation, although this is, of course, arguable because many of us who work in the field consider actually the formation of the first peptide bond as the start of the elongation process. Now, to avoid confusion, let's just stick with what your textbook says about the formal start of elongation as the ribosome movement, or technically called translocation. Okay, in this particular stage, once the first peptide bond has been formed, the ribosome then shifts one codon to the right, as shown here, which moves the uncharged tRNA. That means the tRNA that no longer has any methionine in it to the E site, this here, okay? And the peptide bearing tRNA shifts to the P site. Now freeing, therefore, the A site for the next amino acid charged tRNA in line to come in, again based on anticodon codon complementarity. Okay, so there has to again be codon anticodon interaction that must happen in this process. Okay, as a result of this shift, the uncharged tRNA in the E site is ejected, and incoming third tRNA is now able to interact again at the A site. So once the third tRNA enters into the A site and positions itself based on anticodon codon pairing, the chemistry happens again in the P site, whereby the growing polypeptide, the dipeptide, transfers to the amino acid that is on the A site. So it is always a movement of the growing polypeptide to the amino acid that is found in the A site. So a covalent bond forms between the amino acid arginine that is bonded to the tRNA, to the A site, and the amino acid valine of the dipeptide bond to the tRNA, bonded to the tRNA P site. Okay, so once this happens, the subunit moves down once again by the process of translocation, and then the elongation cycle continues by repeating steps D and E, which is the accommodation of the A site, empty A site with the fourth tRNA that carries a charged amino acid with complementarity of the anticodon with the next codon found in the A site, and then followed by the formation of the peptide bond, and so on and so forth. So based upon your understanding of the codon-anticodon interaction happening during the process of elongation in translation, I would like you guys to answer this following question in your Reef. So again, open your Reef account and answer this question. So referring to the following mRNA sequence, 5 prime, this sequence, what would the anticodon look like if the second nucleotide of the fourth codon was mutated to U? So what would be the consequence of that? So please have a look at the four choices and see what would make sense in terms of complementarity of base pairing between codon and anticodon. So all process, no matter how good and how elegant, must eventually come to an end. And so this is, of course, true with translation. So the elongation process continue until one of the stop codons is encountered. So remember the genetic code that we discussed in the first video. So three of them codes for a stop codon, that is UAA, UAG, or UGA. So if any of these codons eventually gets encountered by the translocating ribosome at the A site, in this particular case, the stop codon that it encountered is UAA, um, no tRNA can recognize this codon, but instead an interesting protein called a release factor. So it's a made of a protein. And this particular protein actually mimics an amazing observation, surprising, but actually not surprising also because it has to mimic the shape of the tRNA. So when they in fact discovered the or uncovered the three-dimensional structure of this particular protein called release factor, um, then they discovered that it has a three-dimensional shape that mimics the shape of a cloverleaf tRNA. And so because of that, it is able to fit into the A site that is normally uh, bound by a tRNA. And because it does not have an active terminus to attach the growing polypeptide then simply gets cleaved and released. Okay, so the breaking of the bond creates the carboxy terminus of the polypeptide, and it completes the chain and simply gets released. And so this ends the process of translation. Once that happens, the large subunit and the small subunit of the ribosome dissociates and gets released into the cytoplasm, thereby terminating the process of translation. So I would like to end this lecture video by coming back to the genetic code and consider the potential of building proteins based on the principles that we have uncovered or studied. We realize that in building proteins, there are in fact, and at least mathematically, an infinite, nearly infinite number of possible sequence that one can build based upon the principle of building proteins based on 20 different amino acid monomers. Okay, so consider, for example, a protein with just about 62 amino acids long, which is rather short for the average protein that we see in cells, which has an average of about 500 amino acid sequence. Okay, so that possibility or possible combination of all random sequences would give rise to 20 to the 62 possible sequences, and that's because every position 1 in amino acid position 1 and amino acid position 2 will give you 20 possibilities. Second amino acid position will give you another 20, and so on and so forth, until you end up with the 62nd amino acid, uh, amino acid 62. Okay, and so that gives you 20 to the 62 possible sequences that can arise, okay, in terms of the uh, maximum possible sequences that you can get. And this is in fact equivalent to the number of estimated electrons, protons, and neutrons in the entire universe. So the question is, why are there no more types of proteins than what we have? In fact, so far what we have seen is that there are only about 25,000 different protein families. So there isn't much. There are a lot of small changes within these protein families, and yet we see that th- there is no large amounts of protein families that we see, but instead a relatively limited amount. And of course, the answer to this question, the simplest answer is the fact that not any random sequence of amino acids in a protein can give rise to efficient function. In fact, the probability that any given random sequence can give rise to a functional protein is very, 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 very close to zero. Okay, so the proteins that we now encounter in living cells are actually a byproduct of an incredible fine tuning in during the process of the development and emergence and evolution of cellular life on Earth, and the role by which translation played in terms of determining the kind of proteins that are able to do um, amazing functions inside the cell is a mystery that still remains to be solved. So this ends this lecture, and I hope that you would be able to appreciate the intricacies of the process of translation in this section. The next subsequent slides simply gives you a summary of the or a synthesis of the process of transcription, RNA processing, and translation as they are found in eukaryotic cells. So let's do a quick summary, therefore, of gene expression as encountered in the segments on chapter 13 and, I'm sorry, chapter 3 and here in chapter 4. So DNA is first transcribed by RNA polymerase in the nucleus of eukaryotic cells to produce complementary antiparallel strand of RNA. And the transcription begins at the promoter region and continues until a terminator sequence is encountered. So all of this is happening here in the nucleus. Now, this RNA is then processed in the nucleus to make mature RNA or mature messenger RNA. And in here, in the nucleus, the introns are excised from the strand, while the exons are spliced together in order to form the uh, um, processed mRNA. And this processed mRNA receives a 5' cap and a long poly A tail, which then travels out the nucleus through a nuclear pore. Finally, the mRNA is then translated in the cytoplasm, as we just described in this chapter. A ribosome attaches to the mRNA through the cap structure and then scans for an AUG start codon. When this codon is found, then it initiates translation and synthesizes a polypeptide chain based on the sequence of the mRNA until it reaches a stop codon. And once the stop codon is reached, the protein detaches and is released for processing. And once it gets processed, the protein can now um, move to its designated location and perform its important function. So that is the overall summary of gene expression, and I hope that this serves as a strong foundation for your understanding of the principles of the central dogma, which is described here in both chapters 3 and chapter 4. Thank you.